Good afternoon to everyone. It's a pleasure to be presenting at the 21st Argentine Congress of Herpetology. This work is entitled First Evidence of Batrachochytrium dendrobatidis in the Fog and was conducted in Brazil at the Laboratório de História Natural de Anfíbios Brasileiros. The group of amphibians is the most threatened, which has been suffering massive species extinctions and population declines since 1989. Currently, more than 40% of amphibian species are included in some threat category. Infectious disease epidemics are one of the main factors threatening amphibian populations. Among the main emerging pathogens strongly associated with the global decline of amphibians is the fungus Batrachochytrium dendrobatidis, also called BD or chytrid, that causes the chytridiomycosis disease, which drove several anurans populations declines and has been found in all continents. Because BD has a flagellated free-swimming zoospore which needs water to survive until infect a new host, it is mainly related to water bodies. So, BD can be transmitted by the stream or lake water, but it also infects amphibians from other environmental reservoirs as soil, plant leaves, and rain contamination. It is difficult to identify BD dispersal routes that occur independently of host amphibians, and it is likely that there are yet uncharacterized mechanisms. Understanding how these transmission mechanisms work is critical to mitigating the disease impacts. So, if BD was found in the rainwater, what about the fog? We can think about the fog as another environmental reservoir of microorganisms. Fog is a collection of water droplets or ice crystals suspended in the air on the Earth's surface. The gaseous molecules of water condense around microscopic solid particles suspended in the air, which can be particulate matter such as dust, pollutants, or microorganisms. So, fog has ecological relevance because it is an important water input in ecosystems. And it carries bioaerosols and microbial communities among them, mainly in cloud forests and coastal environments, causing long-distance dispersal, which is super important, so keep this information in mind. Also, considering that most microbes prefer aqueous environments to survive, fog is an interesting bioaerosol transport mechanism that can provide suitable conditions to zoospores, such as high humidity and low temperatures. Detecting and characterizing the occurrence of a pathogen is relevant to identify the environmental conditions that lead to outbreaks, to develop control measures, and to prevent epidemics. In this context, we asked, can BD be passively transported through the fog? So, the aim was to investigate the presence of BD DNA in the fog. To answer this question, we sampled fog through a passive method using a fog catcher that comprehends a structure with a net. I will show it soon. And we also sampled fog through an active method using a portable aspirator with a sterile filtration membrane. About this active method, we made an aspirator experiment to evaluate its effectiveness. We also sampled rainwater as a control to investigate BD DNA in this reservoir separately from fog events. All these steps generated membranes that were subjected to DNA extraction and qPCR analysis. Between March 2020 and February 2021, we conducted field sampling in 7 areas in the Atlantic forest, in the southeast Brazil, a hotspot of the world's anuran amphibians diversity. Here, Figure A shows the forest Atlantic biome. The elevated areas we sampled were nebular forests, where the fog occurs frequently. Figure B is the mesh we used in the nets we call fog catchers, that we can see in the figure C and D. These fog catchers collected the fog in form of water. Figure E shows the filtration membrane attached to the aspirator, and for rainwater samples, we used a rain trap composed of 2 buckets suspended 1 meter above the ground, attached to a tripod, as we can see in the figure I. So, all the water samples were vacuum filtered, and the membranes were submitted to DNA extraction and qPCR analysis. We experimentally tested the capacity of the portable aspirator method to detect BD in a simulated fog that contained known BD concentrations. We nebulized BD concentration solutions of 10, 10 to the power of 2, and 10 to the power of 3 zoospores per milliliter, which were aspirated for 5 and 10 minutes. We compared the nebulized zoospores quantity with the detected by qPCR. About the results. Out of the 46 fog samples, the results showed a prevalence of 2.2% with 7.3 zoospore genomic equivalent. The BD positive sample was collected in November 2020 in the rainy season at the Parque Estadual da Serra do Mar, Núcleo Santa Virgínia, along the outer edge of a nebular forest patch. Out of the 41 rainwater samples, results showed a prevalence of 9.8%, with loads varying between 4 and 44 zoospore genomic equivalents. All of the BD positive samples were collected in 2019, in January and in April. All of them were collected in the Parque Estadual Campos do Jordão. About the aspirator experiment, for each zoospore nebulized, 0.5 zoospore is detected. With more time of nebulization, the zoospore detection was greater. The BD concentrations detected in experiment were lower than the real nebulized concentrations. However, it demonstrated to be an effective method to detect BD in the simulated fog. From all of that, what can we conclude? Although it is not known if the positive result corresponds to live zoospores, that is, capable to infect hosts, this result provides the first evidence of BD DNA in the fog. This finding supports further studies to better understand if fog configures a pathway of BD zoospores transport and consequent influences chytridiomycosis dynamics. So further studies should focus on zoospore viability. The aspirator is an effective method for fog sampling and, in fact, more effective than the fog catcher method. Our results corroborate the findings of Kolbe et al., adding evidence that airborne transmission of BD can occur. Passive transport is relevant in this process and may reflect new pathogen dispersal routes. We found low BD concentrations in both fog and rainwater, accordingly with what was expected. As the greater the distance from the source of dispersion, the lower the concentration of propagules. However, this also indicates that greater sampling effort must be employed. It is common that cell aggregates can be transported by fog. So, with amphibian skin desquamation, which is a clinic sign of chytridiomycosis, it is likely that viable zoospores can be passively transported through cell aggregates. Finally, from everything that has been said here, I would like the remaining idea to be that there is BD DNA in the fog, and the passive transport of BD by fog may play a role in long-distance spread of the pathogen, which in turn may have many implications for the dynamics of this disease that threatens amphibians worldwide. We would like to thank the agencies FAPESP, CAPES, and CNPq for the financial support, the Universidade Estadual de Campinas for the institutional support. We thank lab colleagues for collaboration and Fundação Florestal for the access into protected areas. And if you liked this presentation and would like to learn more about our research, to discuss new ideas or collaborate, please let us know. You can contact us on Instagram at lanabi_unicamp or by email. Thank you all for the attention.
Gracias por la presentación. No sé si... si hay preguntas.
Bueno, si no hay preguntas...
Sí, está prendido.
¿Está prendido?
Sí.
¿Se escucha? Si no hay preguntas, yo te querría hacer una pregunta, Joelma. Hace poquito me tocó leer un trabajo que muestra que la incidencia del quitridio puede diferir significativamente en las mismas especies en climas diferentes. ¿A vos te parece que este modo de dispersarse puede tener relevancia o relación con esto? ¿Entendiste?
Você pode repetir, por favor?
En inglés, soy como Tarzán en inglés. Hace poco me tocó leer un trabajo que mostraba que la incidencia del quitridio podía ser muy diferente en especies similares, en las mismas especies de anfibios, habitando climas distintos. Entonces, si a vos te parece que estos hallazgos de ustedes pueden ser significantes en relación a explicar este fenómeno.
Bom, eu acredito que outros fatores vão estar relacionados, como fatores ambientais, né, como a temperatura e a umidade. Mas o que nós acreditamos que a neblina pode influenciar a transmissão numa escala regional onde ela vai ocorrer, principalmente em áreas mais elevadas, onde a gente vai ter uma maior umidade relativa e baixas temperaturas. Outro fator que nós pensamos, talvez, é que a neblina pode ter um papel na transmissão da doença para espécies de desenvolvimento direto. Espécies de desenvolvimento direto que não tem a fase larval de girino e tem um menor contato com corpos hídricos.
¿Y no han pensado estudiar la incidencia del hongo en una especie donde la incidencia es alta en sitios con neblina y como tener una especie de control negativo en sitios sin neblina?
É interessante essa possibilidade. Esse trabalho que eu acabei de apresentar foi o primeiro passo, né, para sabermos se há UBD na neblina, e nós sabemos agora que sim. Então, a partir de agora, nós vamos focar em estudos que estudam a possibilidade de infecção né, através da neblina, estudos laboratoriais, e o terceiro passo é fazer um estudo em campo, como esse, por exemplo, que você sugeriu, para fechar diferentes linhas de investigação para conseguir responder essa pergunta.
Muchas gracias, muy interesante.
Obrigada.
Muchas gracias, Joelma. Ahora vamos a seguir volvemos.